Hi everyone, my name is Disliv Kanku, and this is my capstone presentation for the Google and Kaggle AI Agents Intensive Capstone Project. Before we get started, just a little bit of background. I am a data and AI engineer at Dana-Farber Cancer Institute, where most of my work involves building data pipelines and building AI systems, ensuring that we provide researchers with the right tools to allow them to make advancements in their research. So, as part of my capstone presentation, I decided to go create an healthcare research agent. So, my project is titled the Healthcare Research Agent, and which is basically a multi-agent system for extracting and synthesizing clinical evidence. I'm using Gemini, PubMed, and Supabase. So, basically, the problem statement and why this matters. I'll be switching between slides. So I decided to go with this project because clinical researchers are drowning in information just from working alongside researchers. I've noticed that, there's thousands of new biomedical papers every week, especially from PubMed. And the tools that researchers use, for example, PubMed's PDFs and spreadsheets, these are just completely fragmented. And this project proposes a new workflow. So one that turns natural language questions into actionable evidence in minutes instead of weeks. So let's get started with the research problem. So first, the core problem is simple. The volume of research is outpace human capacity to process it, where there's so much research and there's not enough human capacity to process that information. There's so much information that gets produced out there weekly, and researchers spend most of the time just triaging abstracts, manually copying evidence, or searching across disconnected tools. And there's sometimes there's critical decisions. For example, critical decisions in healthcare, critical decisions in oncology, rare decisions, and emerging therapies depend on timely, accurate synthesis. Yet the existing process is painfully slow and inconsistent. So my system is built strictly for research workflow only. So there's no PHI, no patient data, and yeah. Now, why did I decide to go with a multi-agent system? Because of four reasons. First of all, research workflows are naturally multi-step and they are high in complexity. So a single LLM is not enough. It will hallucinate, lose structure, or miss steps. So instead, the system uses specialized agents, each with a narrow verifiable role. So first, we have a query interpretation agent. We have a PubMed retrieval agent, evidence extraction agent, synthesis agent, and this basically makes the system modular, transparent, and observable, and safer than just a single black box. So on the next slide, let's dive into the system architecture. So here's the full architecture. So we have the user, fast API, orchestrator, multi-agent pipeline, Supabase, Gemini, and PubMed. So basically, the orchestrator breaks down the workflow and routes tasks to the right agent. And Supabase stores sessions, logs, and evidence so the entire process is reproducible and auditable. So we have several agents. Let's dive deeper into the agent roles. So here I'll quickly walk through the different agents that we have. So first we have the query agent, which takes a raw question and produces a structured PICO, which is optimized PubMed search query. And then we have the search agent, which runs the PubMed searches, fetches metadata, and prepares the PubMed IDs. We have the evidence extractor agent, which uses Gemini 1.5 Pro to extract interventions, outcomes, effect sizes, and structured findings. And then we have the summarizer agent, which generates a clean, concise research brief with inline citations. We also have the orchestrator agent. This basically manages the agent workflow. So it coordinates message between all agents using a simple agent-to-agent protocol. So this modular approach gives full transparency into each step. So here, this is the deep dive into the query agent. So this is a quick deep dive into the query agent, but which I explained in the previous slide, where we kind of went deeper into how each agent behaves and performs. and we have the evidence extractor agent, the summarizer agent, and this is the data pipeline workflow. Basically, this shows how the system goes from PubMed to extraction to Supabase and to summary generation. So everything is logged and everything can be replayed. So I think now it's time for a real life demo of our platform where we can actually get to see it work. So now I will run a real life demo. So first you start off by creating a session. So we created a session. Let's start with the question. First, let's what adjuvant therapies, sorry, there, there, what can adjuvant therapies, what adjuvant therapies improve OS glioblastoma. So this is our first prompt. So basically what happens, so the user inputs a query. And first, we start off by creating a session. We have to create a session. That's how we're storing memory. That's how we're storing context. And then secondly, the user inputs a query. So inputs a question. So in here, the system will automatically perform five things. The first one is PICO generation. So from the user query, the system puts it in a structure, in a search query structure, which aligns with PubMed, and it searches on PubMed and extracts abstracts. That's the third phase where from this user query, abstracts are extracted, and then we have the fourth agent which extracts evidence, and the fifth agent which will summarize the response with PMIDs, with PubMed IDs, and usually this whole process normally takes hours, but because of our agent, the agent takes just a few minutes. As we have, you can tell, we have a whole research brief from just the first question. So it gives a whole summary, background and question, study characteristics, key findings, references, conclusion. It also shows which tools were used, PubMed, evidence. Uh, it also provides citations to some of the research articles, some of the abstracts which were used in creating this research brief. We can click on this one, and it takes us to the article, to the abstract. So yeah, so this is what we have so far, and this is our live demo. So now I'll be switching back into the main present. So that was the demo of our platform, of our agent working, where we have several agents which are working together in a multi-agent system. Some of them are running sequentially, and some agents are running parallelly. And let's discuss observability and evaluation. So observability and evaluation, these are already built into a system. So we have latency metrics, we have tool usage, we have error traces, we have extraction accuracy. These are some metrics which were investigated, which were constantly paying attention to. Um, so this is an evaluation data set of 10 to 30 curated uh, queries where we have 92% round call, 87% precision, and 89% F1 on structured extraction. And in terms of the tech stack, we use Gemini, Fast API, Supabase, PubMed. We use Docker and Cloud Run and React to build the front end. Everything is built like a real clinical research tool, so it's reproducible, auditable, and secure. Next slide where I'll be discussing security and compliance. So because this is a research only system, so there's no PHI which ever enters the system. There's Supabase role level security which has been enforced. Our API routes are protected and all inputs are sanitized. So this aligns with best practices for healthcare grade AI platform. The next slide, this dives deeper into future roadmap and what expansions looks like. So future expansions include full text PDF extraction, RAG-based citation linking, domain-specific agent variation, and critique loops for self connection. So basically, this lays the foundation for next generation research assistant, which we will be building on, just expanding more on the project. So yeah, I believe that's all. That's all for the presentation. So this project demonstrates that multi-agent AI system can meaningfully accelerate clinical research and make biomedical evidence more accessible. So I'm actively still evolving the systems and documenting everything that I'm learning as I build. And yeah, thank you for allowing me to be part of this program.